So we're in the process of trying to get some more chairs, and hopefully that'll work out. But on behalf of the BYU Gerontology program, I welcome you to the 34th Annual Russell B. Clark Gerontology Conference. My name is Ray Merrill, a faculty member in the College of Life Sciences and director of the Gerontology program here at BYU. Russell B. Clark was an amazing individual. His longevity, he lived to be, was it 105? 109. Lived to be 109. His optimistic personality and his generosity are greatly appreciated. And so this conference is named after him. And he also, his family has provided funding for this conference. So we really appreciate the Clark family. We're going to begin with an opening prayer by Lindsay Barlow, a Gerontology Service Award recipient majoring in exercise and wellness. And following her prayer, we'll hear from a choir, and some of the choir members are recovering from strokes. And so we really appreciate you coming tonight and look forward to hearing you. After the prayer, Tyson Harmon, an assistant professor in the Department of Communication Disorders, will then present the Distinguished Service in Gerontology Award.
Well, I want to thank the Neuroharmony Choir. Let's give them one more hand. And I'm thrilled to be able to introduce this year's Distinguished Service Award recipient. Tyler Hewish is a dedicated physical therapist at Utah Valley Hospital with a passion for helping older adults regain independence after stroke and other neurological conditions. Throughout his career, Tyler has focused on rehabilitation and recovery, providing expert care and support to patients and their families. Beyond his clinical work, Tyler is the founder and first president of the Utah Valley Stroke Association, a nonprofit organization that offers resources, education, and activities for leadership and, for stroke survivors and their loved ones in the Utah Valley area. His leadership and commitment to this cause have made a lasting impact on the local community, creating a network of support for those navigating life after a stroke. Tyler's tireless dedication to serving older adults, particularly stroke survivors, has made him a respected leader in his field. His compassionate approach and advocacy efforts continue to improve the lives of countless individuals and families in the region. On a personal note, I've had the privilege of serving on the UVSA board with Tyler, and I can say from personal experience that he is completely dedicated to stroke survivors and their families and to helping them have social opportunities and experiences that will help them throughout their recovery. I'm very grateful for the work that he does. So please join me in congratulating Tyler for this year's Distinguished Service Award.
I still remember as a little boy, I was in a red Toyota with my mom driving from Farmington to Salt Lake, and I heard that song, Sweet Caroline. And I loved the song, and I thought, I want to marry somebody named Caroline when I get older. But I didn't. In fact, I don't know if I've ever met anyone named Caroline, but it's a great song. And congratulations, Tyler, for that award. So our keynote address will be given by Dr. Ben Bikman, who will present on the metabolic origins of cognitive decline. Dr. Bikman is a professor of cell biology and physiology at BYU, whose research examines the molecular mechanisms that explain the increased risk of disease that accompanies weight gain, with particular emphasis on the etiology of insulin resistance and disrupted mitochondrial function. You can read about Dr. Bikman's experience and research in your program, but he's had extensive experience in research and presenting around the world. I often see him down in the locker room, and he's telling me down in the Smith Fieldhouse, and he tells me he's just getting back from someplace like the Middle East or Tokyo, or where was your last trip? Thailand? Yeah. He's always off somewhere, so we're really fortunate to have him here. He's a good friend, and let's welcome him.
Thank you. I am delighted to be here. What Ray didn't mention is that he actually offered the invitation to speak while we were in the locker room. So a lot happens there. It's a magical, productive place. Let me just start by expressing my affection for BYU, how I love this university, and as much as I am invited to share a portion of this message in a variety of places, none compare to BYU. I love what this university stands for, and if it weren't for that, I would perhaps be somewhere else. But I'm delighted to be here. I was delighted to have an opportunity to put together a message that I haven't given yet, which is a particular focus on how metabolic disruption compromises the brain, how really this is the new paradigm that I'm about to share with you, the bleeding edge of science when it comes to where Alzheimer's research is going. Now let me just set the stage, and it's a sobering one when we look at the statistics, that up to 88% of US adults are considered as having an unhealthy metabolic state, where this study noted, and I'm sharing the quote, that even in normal weight individuals, there's an alarmingly low level of optimal metabolic health. Now, just by way of reference, you'll notice in the corner of many of my slides, I have a little PMID. That's just so that you don't think I'm making something up. That is for you to go and do some of the homework on your own and look up the peer-reviewed manuscript that I'm citing. Now, as Ray mentioned, as much as we have a problem here, it is a uniquely American trait to think that we're the worst at these kinds of things. I say that as a foreigner. We Canadians blend in so well, don't we? But this is not an American problem that, as Ray mentioned, I've given some version of this talk. In other words, the metabolic origins of chronic disease. This is just last year, actually. In fact, I'm not even showing all of them. But if you look across South Asia, East Asia, the Middle East, that is the hotbed of metabolic problems. In fact, the statistics are even worse there than we have here. Throughout the Pacific Islands, I once was told in a regional church meeting that the most common reason to have to reorganize a stake presidency is that someone dies from type 2 diabetes, which, as you'll learn, is just a problem of insulin resistance. And even in our own neighborhood, Mexico has worse rates of metabolic problems than we do. So this is a global problem. Now, I've been saying and indeed invoking statistics that look at metabolic health, and yet as a scientist, that's pretty imprecise. I like my language to be as precise as my haircut, which is no room for mistakes. What does metabolic health even mean? One way of defining metabolic health, and indeed the most common way that is cited in peer-reviewed scientific manuscripts, is to define metabolic health as nothing more than the metabolic syndrome. Now, those of you who have been going through any health-related classes or not have probably heard this term before. It is a cluster of complications that just tend to clump together. You always see these things just cropping up and going hand in hand, and you see them indicated here. Now, even still, for a person who is seeking precision, this is not precise enough, because the metabolic syndrome sounds very nice, doesn't it? We just have such an interest in the word metabolic and metabolism, and yet it doesn't tell you what these things have in common. We then have to go back in time, because before we called this the metabolic syndrome, we referred to it as the insulin resistance syndrome. And Dr. Gerald Reaven first identified these problems that always tend to come together. He called it Syndrome X, then the insulin resistance syndrome, And that just wasn't provocative enough, I suppose. And then they called it now what we refer to it as the metabolic syndrome. But if we are hoping to help people understand what all of these seemingly distinct problems have in common, we understand it better if we call it what it really is, namely the insulin resistance syndrome. So that allows me to introduce the villain of the story. And as sobering as the statistics are with regards to the prevalence of insulin resistance, it gets even more sobering when we view the relevance of it, which is how causal it is in virtually every chronic disease. So the main killers, the diseases that are wreaking such havoc, what I like to refer to as plagues of prosperity, you wouldn't think type 2 diabetes and heart disease necessarily have a connection, or what would be their connection to fatty liver disease? Well, they're all to various degrees directly caused by or exacerbated by insulin resistance. But the problem goes further than just these big killers, from the perhaps less lethal but the perhaps more heartbreaking problems of fertility are also derivative of insulin resistance, where the most common form of infertility in women, polycystic ovary syndrome, is increasingly referred to as metabolic infertility. Rightly so. Also, erectile dysfunction, the most common form of infertility in men, is considered to be one of the earliest manifestations of insulin resistance in an otherwise healthy fella. So even infertility in its most common forms is also derivative of this metabolic problem, insulin resistance. And then we come to the problems of the brain, which I promise I'm going to get to, but problems like dementia, migraines, even depression. I'm going to show you in the next few minutes what these have in common, other than they're all manifestations of a brain disorder. It actually goes a little deeper than that. Okay, so you know how common insulin resistance is, and you also know how it influences all of these diseases of civilization, and yet you don't actually know what insulin resistance is yet. So let's take a moment to define it. Insulin resistance is most simply understood as a pathology with two parts. The first part is the one you think of when you look at the name insulin resistance. Namely, you think the hormone insulin isn't working particularly well at various cells of the body. And this is relevant and noteworthy because insulin is unique within its family of hormones in that it affects every single cell of the body. From brain cells to bone cells, from lung cells to liver cells, and every cell in between, insulin will tell that cell to do something. So some cells aren't listening. They've become like a naughty little child, and my wife is here and she understands that analogy all too well. Now, that's not the end of it though. As I said, there's two parts, so we have to flip the coin over in order to really understand how insulin resistance is so harmful, and that is that insulin levels are elevated. There is no such thing as insulin resistance without this hyperinsulinemia or this chronically elevated insulin. They always come together. Now, let me adjust the paradigm a little bit and present it in a way that is commonly discussed perhaps in a clinical conversation, which would be, imagine a patient coming in every year for a wellness visit. And during each wellness visit, among the many blood markers they look at, the singular marker that is relied upon to get an idea of metabolic health is fasting glucose. And the fasting glucose levels may look something like this, where for many, many years they're normal, and then eventually for several years they climb. Now, in a discussion about insulin resistance, you'll notice that something is absent. And if we superimpose insulin on this graph, it starts to tell a very different story. There's actually a lot of wisdom to be gleaned from this one single slide, if I do say so myself. In fact, it fundamentally challenges the view of type 2 diabetes, which I'll get to in just a moment, because that's what insulin resistance gets to. But you'll notice that there's really two distinct time points on this lifespan of the individual, where there are a number of years where glucose levels are staying normal, but it's taking ever more insulin to keep the glucose levels normal. This is insulin resistance, or as some refer to it as pre-diabetes. If you've ever heard that term, it is synonymous with insulin resistance. But it is only once the body becomes so resistant to insulin, whose most famous effect is to lower blood glucose, that now it can no longer control the blood glucose, and then we start to see a state where blood glucose levels are climbing, and now it reaches a point of clinical significance based on the conventionally trained clinician. They say, oh, now your glucose is climbing, there's a problem. But can you see the issue? Our glucose-centric paradigm actually has us looking at the wrong marker. Glucose is not the best marker of insulin resistance. It is insulin, the petite peptide or the humble hormone that is overlooked even in its own story. It's trying to become the main character, and yet we continue to treat it like Robin when it's actually Batman. So we have to measure insulin in order to detect insulin resistance. In fact, glucose levels won't start to climb in many people until the insulin has been elevated for 10 or 20 years. Imagine the opportunity to intervene if we detected the problem at the earliest possible signal. Now, where does insulin resistance come from? That's the final part, really, of this before we transition into the brain more explicitly. But here, insulin resistance has multiple causes in what I often refer to as fast insulin resistance and slow insulin resistance. I'm only going to spend time talking about the one that's common between the two, and that is chronically elevated insulin. Now, some of you are really paying attention, and you're thinking, hold on a minute, though, Ben. You just said that high insulin is a feature of insulin resistance. Yes, and I'm saying that it's also the primary cause of insulin resistance. So you can begin to see how this becomes a vicious cycle. But too much insulin causes insulin resistance. This is, in fact, fundamental, reflective of a fundamental biological principle, which is if a cell is inundated with a signal, it will attempt to become resistant to that signal in order to maintain normal function. So living a life of chronically elevated insulin will drive insulin resistance. As insulin resistance progresses, the body needs more and more insulin in order for the insulin to get its job done, which simply exacerbates the insulin resistance. Now, how has this become so common? If elevated insulin is the main driver of insulin resistance, and insulin resistance is the most common health disorder, naturally we should understand, well, what drives up the insulin in the first place? Well, it is the food we eat. Now, what do we eat? We have 3 macronutrients, the large part of our diet: fats, proteins, and carbohydrates. Well, I've given versions of this talk globally, so I am sort of a globally minded individual when it comes to this problem. And globally, over 70% of all calories consumed come from carbohydrates. That's, of course, telling, all the more so when we consider what each of these macronutrients does to the hormone insulin. When a human eats pure fat, there's no insulin effect. Now, most people, it is an uncommon roommate, right, kids, that is eating a stick of butter while you're watching Netflix or something. But it is people, you can eat pure fat, and there will be no insulin response whatsoever. You can eat pure protein, which many of you young men do, in fact, so obsessively that I almost imagine you snorting little lines of whey protein powder off your kitchen counter. So we have we eat plenty of protein, and when a human eats protein, there's no insulin effect, or little to no insulin effect. There's a little bit of nuance there. But this is actual human data. You can see if there is an effect, it's quite modest. When a human eats pure glucose, which is sort of the most pure form of carbohydrate, if you will, there is a substantial increase in insulin. Remember, this is actual human data. I'm just presenting it with a particular aesthetic. I know it's funny for a guy who looks like me to have a strong feeling of how things look, but I do. In fact, my wife is the one who doesn't have a strong sense of aesthetic, if you know what I'm saying. So here we are. But here we see what the insulin response is to pure glucose. Now, importantly, this is what insulin looks like when a person is insulin sensitive. Remember, that is the uncommon adult nowadays, where most adults have some degree of insulin resistance. And if an insulin-resistant person were to eat that same amount of glucose, the insulin response would not only be actually multiples higher, so this is now not to scale, but it would also take much longer for the glucose to come back down, the insulin to come back down. The insulin could be elevated for up to 3 or 4 or even 5 hours before it ever has an ability or an attempt to come back down to baseline. This matters, in my last sentiment here before talking explicitly about Alzheimer's disease because of how we eat. So I mentioned that the most common macronutrient is carbohydrates, overwhelmingly, and we eat them so often. So this is what it might look like, insulin levels in an average individual who just eats 3 times a day. In fact, that's not average because most people don't eat 3 times a day. We now have a culture where we have to fill in any potential gap. We eat a big hearty breakfast, and then an hour or two later we need our mid-morning snack, and then we need an afternoon snack, and oh my gosh, don't deprive me of my evening snack. And so an individual is spending every waking moment in a state of elevated insulin. And all the more so if a person has insulin resistance. Then it takes so long for the insulin to start to come down that it never even has the slightest chance. So every waking moment is spent in a state of elevated insulin. And you young students, I know what you're going to do. You're going to go home and eat 3 bowls of cereal or something. And so most people are even spending several hours into their nighttime, into their sleeping hours with elevated insulin. And as an aside, the worst possible time of the day within a 24-hour period to spike your glucose is before you go to bed. But that's a topic for another time. Ray will have to invite me back another year for that talk. Okay, now what about dementia? Let's actually get to where the rubber meets the road here. I don't need to paint this sobering picture when it comes to the statistics of Alzheimer's disease. It's a terrible disease. And we all have, I suspect, a very healthy appreciation for this problem. The classic view of Alzheimer's disease is that it is caused by plaques in the brain. But let's think like a scientist, and a scientist is nothing more than a seeker of truth. Are the plaques causing the disease, or are they simply accompanying the disease? There are 2 problems with the plaque-based view of Alzheimer's disease. One is that there are many, many people who die with Alzheimer's disease and have no plaques whatsoever. And second, that there are drugs that reduce plaques in the brain, and yet they don't improve cognition at all. Let's just review them very briefly because I really need you, in order to appreciate the metabolic origins of Alzheimer's disease, you have to put to bed the plaque-based view. So this was a paper published within the last 10 years or so, where they noted, looking at post-mortem biopsy samples of the brain, not a biopsy in this point, they're dead. Thankfully, it's difficult, although a student will do anything for $200, but maybe giving a brain biopsy would be asking for a little too much. But here, they took brain samples from people who had passed away, having died with strong evidence of Alzheimer's disease versus no evidence whatsoever and were cognitively perfectly sound. And they were just as likely to find plaques in either set of brains, that there was no rhyme or reason to it, much like attempting to determine that LDL causes a heart attack. Well, just as many people are going to have a heart attack with high or low LDL cholesterol levels, another problem with modern medicine. The second problem, and this one's a little more intriguing, is that there are drugs that reduce plaques very well, and they don't help. In fact, let me tell you a funny little love story when it comes to anti-plaque drugs. In 2020, the FDA called a special panel of experts in the Alzheimer's space to review a drug that was seeking approval. And the drug was marketed or wanted to be marketed as an Alzheimer's drug. And it was used to help reduce plaques. The special panel of experts on the topic reviewed the drug and all of the data, and they said, well, the drug actually reduces plaques, but it does not improve the disease at all. And combined with that, it's extremely expensive, so we recommend that the FDA reject approval for this drug. The FDA then said, well, even though it reduces plaques, and so we think it will likely improve cognition, even though the experts, having reviewed the data, said it does not improve cognition. In fact, the FDA went on to say, we don't want to reject this drug for fear that it may discourage further drug development, even though it doesn't work. And so they overrode the panel and approved the drug anyway, and it went to market. Making this particular view of Alzheimer's disease all the more tragic is that in its very origins, it appears to have been based on fabricated data. So indeed, the very first seminal manuscripts that gave birth to the plaque-based theory of Alzheimer's disease were based on fabricated or faulty data. And this made headlines a couple years ago, and so it calls into question the plaque-based theory of the disease from its very origins. Now, this was very gratifying for those of us who have been saying that the plaque-based theory is weak and that there's another view that needs to be considered more seriously, and that is the metabolic view, or what I'm calling fuel disruption. Now, I have the flame there not to suggest that the brain is burning, but rather that it has a high metabolic rate. The brain actually occupies a trinity of high metabolic rate organs, with the kidneys and the heart, and then the brain is next most busy. In fact, the kidneys are unlike anything else in the body. Their metabolic rate is so fantastically high that when my students attempt to measure the metabolic rate, it uses its own entire separate protocol. But the brain is a high metabolic rate organ. It is a busy organ. Hopefully, it's busy now as you're learning. So it needs to be fed. Like a high-performance engine, it needs to have a consistent, good source of fuel in order to operate very well. One such fuel, and indeed the only one most people know about, is glucose. But there is an alternative fuel. The brain is a metabolic hybrid, and it also can rely on ketones. Now, these are not the same. Now, even uttering the word ketone can be very upsetting for some people. And unfortunately, it's very unfortunate. Ketones have been considered metabolic garbage for decades, and it is an incredibly unjust perspective. Ketones are actually able to get into the brain very readily, unlike glucose, which in certain regions of the brain, including those involved in cognition and memory, require some degree of insulin in order to knock on the door of those neurons, open the doors for the glucose to come in. So in that regard, insulin is acting like a bit of an usher. Again, coming to the cell, with seeing the glucose, saying, glucose, you want to come in and feed this brain cell, I'm going to knock on the door to let you in. The glucose can't go in on its own. Unlike other tissues are able to pull glucose in without the need of insulin to mediate that process, but not certain sections of the brain that are very important for cognition. Now, I've mentioned a new character in this story, which is ketones. So let me go on a brief tangent and explain to you what ketones are. As much as I just presented the view that the brain is a metabolic hybrid, primarily relying on glucose and ketones, the body, by extension, is also a hybrid which primarily relies on glucose and fat. Now, there are other fuels, of course, like ketones, like lactate. And by the way, humans don't make lactic acid. There's no such thing in humans. But we burn lactate very readily as a fuel also. But the main fuels are glucose and fats. Well, what determines which fuel is being used? It is, once again, the humble hormone insulin. When insulin is down for some period of time, and in a non-type 1 even in a type 1 diabetic, if it's at very low levels for roughly 12 to 16 hours, it will have been burning so much fat as low insulin shifts the metabolism to fat burning that it will start creating these byproducts of fat burning, and that is ketones. So just to demystify and somewhat, I hope in the next few minutes, devilify ketones, they are nothing more than products of fat burning. So that brings us back to this view. Most of you students have been taught the tragic perspective that the brain can only rely on glucose. And a professor might have even cited a specific study, because in a fed state, like those of you who are just up at the dinner on the third floor, if we are eating a meal of mixed macronutrients, proteins, fats, carbs, that high glucose will be the singular fuel for the brain. So in the fed state, which is about 4 to 6 hours post-meal, depending on what you eat, the brain is relying on glucose as a fuel. However, once the person starts to enter the fasted state, now up to 75% of all of the fuel in the brain is coming from ketones. Let me just put this in perspective, although I didn't intend to take the moment to explain this. With a 24-hour fast or so, what Dr. George Cahill, who was the pioneer in this realm, he is in the realm of physiology he's a legend, which means to say nobody knows about him but me and a handful of other metabolic scientists. When a human was fasting between 24 to 48 hours, glucose levels would come to a level of about 5 millimolar, and ketones would get to a level of about 3 millimolar. So less than the glucose, and already in this state, the brain had shifted over so that it was using 3 times more ketones than it was glucose for its energy. So as much as people want to say the brain prefers glucose, I just proved it wrong. Even at lower levels, the brain has shifted to rely on ketones as its fuel. So if the brain prefers any fuel, it's the ketones. So in the fasted state, the brain has shifted. Now, how does this connect to Alzheimer's disease? Dr. Stephen Cunnane in the far northern reaches of Canada, actually in northern Canada, so that's saying something, that's northern squared, he found that in cognitive decline, in patients with early stage Alzheimer's disease, there was a significant reduction in the brain's ability to pull in and metabolize glucose. So this is human level data with some beautifully conducted studies. He's not only an incredible scientist, he's also just an absolute gentleman, a wonderful guy. And we've collaborated and met before. In fact, he gave a talk at BYU years ago. And so what else, though? Looking at the other fuel, while the cognitive declined brain was compromised in its ability to get glucose, it was totally uncompromised in its ability to use ketones. Now, the tragedy is, of course, that the brain may not have access to ketones because it depends what the person is eating. Now, my lab followed up on that particular project. Funding that we received in part from the Gerontology Center here, we were able to get brain biopsy or brain samples post-mortem from cadavers. So this is human data. And we actually did a survey of genes related to glycolysis, so the degree to which these genes are expressed that result in glucose burning, and compared them to the genes involved in ketone burning. And we found that every gene related to glucose metabolism was significantly down-regulated in the brains of people who had died with Alzheimer's disease, and yet the ketone-related metabolism genes were still normal. We also published another paper, again in part funded by the money we received from the Gerontology Center, where we actually looked at rodents, where you can take brains from subjects, and we found that in the animals that were getting ketones in their diet, they had improvements in their memory, and the mitochondria were working more efficiently. In other words, creating more energy for every little bit of oxygen that the mitochondria were using. Now, just to round this out with a little more data that go beyond my own lab, a paper was published in Scandinavia, which the beauty of Sweden, for example, is that they have such clear documentation of patients and citizens and the health records. And they were able to do a prospective study and look back in time at people who were diagnosed with Alzheimer's disease and then look back decades to look at their markers and follow them year after year. They found that there were 8 statistically significant variables that appeared to be predictive of who ended up getting Alzheimer's disease. 3 of them aren't particularly surprising. Age was a variable. In other words, the older the person was, the more likely they were to get it. That's a bit underwhelming. Education was a variable. The more education a person had, the less likely they were. And then the ApoE4 genotype. Now, that's probably one you've heard about because the ApoE4 gene can encode for the ApoE4 protein that can contribute to those plaques that are so dreaded, even if perhaps unnecessarily. But there is a relevance here that can't be ignored. But even then, the metabolic view can explain it because the ApoE4 protein actually can bind to the intracellular domain of the insulin receptor and make it not work very well. So even ApoE4, it's not a plaque story, but it is still an insulin story where it compromises the insulin signal, which then makes the brain start to go hungry. Now, back to this study, you'll note that I mentioned there were 8 variables. Well, what about the other 5? Every one of the other 5 variables was related to glucose metabolism or insulin resistance. So this is part of what has led to the new term for Alzheimer's disease, which is type 3 diabetes. Now, I don't personally love that term. It's imprecise, and I like precision. It's nothing more than insulin resistance of the brain. It is not a different type of diabetes. I appreciate what these authors are wanting to say, although I think it leads to more confusion. This is simply more evidence of insulin resistance of the brain. So when the brain wants glucose as a fuel, if insulin isn't working, then we have a problem. Now, what about the evidence on ketones? This is probably among the highest funded areas of research right now within the Alzheimer's realm, but some early reports have been very positive, like using coconut oil, which is a very ketogenic fat. I actually had a series of other slides, and I don't regret taking it out because I always take longer than I intend, where I looked at different fats in the diet, long-chain fats, medium-chain fats, and short-chain fats, and presented the view that medium and short-chain fats are unique because they cannot be stored. The body has to burn them. And when you're burning a lot of fat, you're making ketones. So this case report found that in patients with Alzheimer's disease, when they were given coconut oil, in the following hours, they saw distinct improvements in their cognition and function. Another paper published finding that when a patient with Alzheimer's disease received a ketone drink, a ketone supplement, that they were able to perform better immediately on cognitive tests. And for example, they reported the ability of the patient to now tie their shoes when they couldn't just an hour or two before they took the ketone supplement. Now, it's not reversing the disease, but it is providing an acute improvement. Now, as I mentioned earlier, the metabolic problems in the brain go further than just dementia, where we also see issues of compromised glucose metabolism in depression and migraines. In fact, the earliest human studies that have been published looking at the use of ketogenic diets actually were published in people with migraines as early as 1910. This is an old area of research. And just to put a fine point on this part of the conversation, the brain, it's like it's drowning in a sea of glucose. And you guys when I was a boy growing up in Canada, if you ever got sent to the principal's office, my principal made us memorize poetry. And one poem I had to memorize once was The Rime of the Ancient Mariner, where the sailor who is stranded on the ocean and dying of dehydration bemoans his fate that he's surrounded by water and he can't drink it. Well, this is a metaphor almost for the brain, where the brain is drowning in a sea of glucose. Glucose everywhere, nor any drop to drink. It simply cannot get it. And this is where the ketones act as, if you will, a life raft, sparing the brain its starvation-induced cognitive decline. But again, the tragedy is, as much as we live in a culture obsessed with consuming carbohydrates constantly, our insulin is always elevated, and the more insulin is elevated, the less we are to ever make ketones. And so we are depriving the brain of the fuel it's crying out for, allowing it to drown. All right, now, for the sake of time, I actually don't know how I'm doing on time. I'm fine. Ray says I'm fine, so you guys just sit tight. Okay. I have enough of my former students here that I have my eyes on you, kids. All right, let's go back to this view, and I'm actually going to use this view to bring in part of the problem with our traditional perspective on metabolic health, because some of the most commonly used drugs to treat type 2 diabetes are drugs called sulfonylureas or just insulin itself. And this is based on the view that the only problem is the high glucose. And so what this conventionally trained clinician is doing is saying, hey, you have high glucose. I don't even know what your insulin is because I don't measure it, and I've never been taught I should measure it, except my students, and maybe the new med school students, I hope. But what they'll do then is say, well, one way to lower your glucose is going to be to push your insulin up. Now, unfortunately, as meta-analysis after meta-analysis has shown, there is no clinical improvement in type 2 diabetics when they're treated with insulin therapy. In fact, what will more insulin do to a person with insulin resistance now that you know what causes insulin resistance? Yes, of course, it will make it worse. This is like giving an alcoholic another glass of wine, hoping it will cure their alcoholism. You're giving them more of the very thing that's causing the problem. So it's no surprise that every health outcome gets worse. Insulin resistance gets worse, as these studies show. I could show you studies on heart disease. They die more from heart disease. They die more from cancer. And they even get more Alzheimer's disease. When you give them insulin therapy, you are driving the insulin resistance, which in turn drives the other pathologies, including of the brain. Now, I would be remiss if I didn't spend at least a moment talking about this new beloved class of drugs, the GLP-1 drugs. Have you guys ever heard of these? They are the famous drugs that I'll get to in a second if I look at what's coming up next. So GLP-1 is a hormone that the gut makes, and it has many roles in the body, but its most famous roles are that it will slow the rate at which food is moving through the intestines to make you have a bulk in your stomach longer, convincing you to eat less. And it will stimulate the brain to promote satiety. In other words, reducing the hunger signal at the brain. So you eat less, which helps your glucose come down, and as your blood glucose comes down because you're eating less glucose, insulin also comes down. So it can improve insulin resistance. Now, one reason I'm sympathetic to these drugs is a paper that was published in 1996, which was a wonderful year to graduate from high school, and also a wonderful year to make a seminal discovery in the realm of GLP-1. This paper took lean and obese individuals and in one instance had them eat a lot of fat and looked at their GLP-1 response. Remember, if GLP-1 goes up, a person's going to feel more full and they're going to eat less. So when the lean and obese populations ate fat, they had a roughly similar degree of GLP-1 increase. When they ate carbohydrates, it told a very, very different story, where the obese group had essentially no increase in GLP-1. This suggests, and it is, I think, powerful, that there may be someone who eats a given amount of carbohydrate and has a significant increase in GLP-1, which tells their brain, you're done. And then they casually push the plate away and they don't need to eat anymore. Then you may have another person who eats that same amount of carbohydrate and they don't get a GLP-1 response, robbing them of some of the satiety signal, and they say, can I have seconds? Can I have thirds? They're not getting the signal telling them to stop eating. So I'm sympathetic to an approach that seeks to increase GLP-1, even if I have some reservations with how they're currently being used. So these are the main drugs. You've certainly heard of these by now. They have become famous. In fact, as a biomedical scientist, I have never seen a class of drug become so embraced and even adored as quickly as these drugs and as substantially. No one brags about being on a blood pressure medication. No one brags about being on a diabetes medication, but they will brag about a weight loss medication like Ozempic, for example. Now, some of my reservations are born from the fact that there are studies showing that some people actually experience a paralysis of their intestines, that it slows their intestines down too much, causing them to die in some instances. It also can result, as this paper found, in a significant loss of lean mass. This report is substantial, where they documented that 40% of the weight that patients are losing is coming from fat-free mass. In other words, muscle and bone. I confess, if you'll allow me to, I have a particular concern for some of you young women who might be tempted to go on these drugs and are already of a healthy body weight, potentially aging your bones, where within a year or two on the drug, you may have the bone mass of a 60-year-old woman, despite only being, say, 20. I'm very worried, to be perfectly frank. Now, let's come back to the brain. The main mechanism of action that people report with these drugs is that it helps quiet the food noise. Have you heard that term used before? The food noise, the constant nagging at you to go back to the cupboard or to the fridge and get that sweet and gooey or salty and crunchy treat. So what is the effect on cravings? This paper documented this, looking at cravings with a survey. And this is an example of what they were surveying on that right side of the screen. How much are you wanting these things? And they found at about 6 months-ish on the drug, there was a significant reduction. Compared to the baseline, you can see that it went significantly to the left, suggesting great improvement in controlling their cravings. At 1 year, it was still significant. At 2 years, it was gone. That the efficacy of the drug in helping them control their cravings had been wiped out. They had become resistant to the effects. Remember what I said about too much insulin? Too much GLP-1 will cause a resistance to GLP-1. And so they had a total return of sweet cravings at 2 years. Now, what about the brain? Because after all, that's what I was invited to talk about. At the brain, there does appear to be some evidence of an improvement. In fact, this, you would say, that's a significant reduction in risk of type 2 diabetes in people who are on the drug. But please look at the language. It's compared to other anti-diabetic medications like insulin or the sulfonylureas that I showed you earlier. So they have the nature, with some statistical shenanigans, of saying, they have the benefit of being able to say that on these anti-diabetic drugs, the risk of Alzheimer's disease goes up. I showed you that a moment ago. And then they're able to report this relative reduced risk compared to that. They didn't report the increased risk compared to baseline. So there might be an improvement here, and I suspect there is, but we have to balance it with those other side effects, like the loss of lean mass and the potential to slow the guts down to a problematic level. But I'm not done talking about these drugs, and I'm not done talking about the brain, because just a couple months ago, a paper reported mental health outcomes. And this further makes me worry about young people in the midst of a mental health crisis. They reported that there was an almost 200% increased risk of major depression on these drugs, and a 100% increase in anxiety and even suicidal behavior. So as much as people say, these drugs help me reduce my cravings for sweet foods, perhaps another way of saying it is, they reduce my cravings for everything. A person who once enjoyed going out and playing pickleball doesn't really enjoy it as much as they used to. A kid who enjoyed coming home from college and playing video games with his roommates doesn't really want to play it anymore. Everything starts to come down by way of enjoyment, perhaps. And this could be why, as published recently in the United States, about 70% of all users of these drugs get off them on their own at 2 years. And what about when people get off? Well, of course, they gain their weight back. Now, you would say, looking here, but they didn't gain all of it back. But what do they gain back? Imagine, if you will, a 60-year-old woman where she's been on this drug and she's lost 50 pounds. Remember, 40% of that was lean mass. 60% was fat mass. If she gets off the drug, one of those masses is going to return very, very quickly. I deliberately chose that sex and age on purpose, because trying to recover muscle and bone as a 60-year-old woman, good luck. So as they gain weight back, it is very, very likely that as a percent of the body weight, she's fatter than she was before she ever started the drug. Of course, it would just as likely apply to a male, but a guy would have the advantage of perhaps gaining some of that lean mass back a little more readily. Now, let me end very briefly. It's going to be a rapid ending, but just some insight from a metabolic scientist. I hope I have impressed upon you the fact that the food we eat is the culprit or the cure. That what we eat and how frequently we eat it is what's driving poor metabolic health, specifically insulin resistance. And as insulin resistance goes up, it contributes to all of these diseases of civilization, or these plagues of prosperity, including Alzheimer's disease, diseases that were once unheard of. So what is my insight? If the food we eat can be part of the cure, I submit that there are 3 central pillars based on the 3 macronutrients. Very briefly, I believe that the first principle should be control carbs. The second should be to, as you are controlling your carbs, prioritize protein and don't fear fat. Now, let me add a little bit to this, because I don't want anyone to leave here thinking, rolling their eyes and thinking that I just simply declared war on all carbohydrates. Not at all. You should consider the source of the carbohydrate. That if these are carbohydrates that come in bags and boxes with barcodes, you ought to beware them. How many B's can I put in a row? Beware of carbs that come in bags and boxes with barcodes, because that is what's going to spike your insulin the most. Whole fruits and vegetables, eat them liberally, enjoy them. Don't drink them, eat them. If God intended us to drink apples, I submit that's how we would pluck them from the tree. And even what we have as fruits and vegetables are fantastically different from what we would have seen as fruits and vegetables 100 years ago. But even still, focus on fiber. If the carbohydrate is coming with fiber, it's generally going to be a better one. And then second, insofar as your meal allows you to, carbs come last. There's some good human studies to show that if you mix around the macros and put carbs at the end, the insulin and glucose response is 50%, about 47% lower than what it would be otherwise. So carbs, put them in their proper place, which is at the end. And then what about prioritized protein? It is inconvenient to admit this, but a scientist doesn't need to be bothered with convenience or social trends. The best protein is animal-sourced protein, especially as we age. A paper was published within the last month that found that older humans need better protein. So as we get older, we need more animal protein. A younger person can waste a little more of their money and gut space on plant protein, but we who are getting up there, we don't have the time for that. We got to focus on the best source. And the best source will always come with fat. There's no such thing as a natural protein that doesn't come with fat. That's how we should eat it. When humans eat protein and fat together, we digest the protein better and we get a better anabolic response. Muscle growth will be better when the protein comes with fat. It's almost as if God knew what he was doing by always putting them together, and in our hubris, we've pulled them apart. But with this view in mind, you will be able to lower your insulin for substantial periods of the day. And with Alzheimer's and cognitive function in mind, I believe this is a very prudent strategy because this is the only effective way to actually make ketones. And I don't ever mean to be seen as an advocate of ketones and ketogenic diets, but when it comes to Alzheimer's disease, the evidence is so convincing from my own lab and others around the world. Ketones are a therapeutic nutrient source for the brain, and we ought to give the brain a break from the incessant glucose and the insulin that is required to get that glucose in. Let your brain have some ketones for optimal aging and cognitive function. Now, in the time we've had together, you are experts on what insulin resistance is and where it comes from with its main source of elevated insulin. You're generally familiar with how it affects the brain and thus cognition. And again, spoiler alert, it's different parts of the brain that as they're affected metabolically manifest as migraines or depression. And then you also have some inkling of what to do about it. And with all of that having been said, I thank you for your attention, and I would be happy to answer some questions if the format allows. Ray? Ray has given a thumbs up. Thank you. Ray, can you empower me just to point to a hand that goes up? Okay, at the back, blue shirt.
Have you noticed in your research any correlation between the animal proteins being fed on the diet of grain versus grass?
Grain versus what? Grass? Oh, yeah, okay, so the question, grass-fed beef versus grain-fed beef. Yeah, so I like that you use the word correlation. I'm a basic scientist, so I hate the word correlation. I want causality. But there's no effective difference. That there's a slight shift in some of the fatty acids, but almost all cattle are only, they're grain finished. That the vast majority of their life has actually been grass-fed and then they're grain finished. And the slight differences, extraordinarily slight differences in fatty acid composition appear to have no effect on human health. So my view is, my view very much is that beef is the single most nutritious food on the planet, to be perfectly frank. And I think that however you get it, enjoy it. Yes, black shirt, mustache, which hopefully your girlfriend will make you shave sometime soon. I'm kidding. You obviously won her with that mustache, so keep going.
I've noticed a correlation with
Correlation?
or a causation.
Okay.
Ketones and maybe an improvement in ADHD or ADD in kids.
Okay. Yeah, so that's a good question. I am only aware of one single case study that was reported in a child subject with very clinically relevant ADD. They adopted a ketogenic diet like many kids do to control their epilepsy. So there are kids who live their whole lives on this. And there were, in fact, reported improvements in ADD. Yes, checkered jacket, ponytail. What were those problems, and what is your data source? I don't know. Yeah, so I'm. Yeah. So I'm not. I tried to make that clear at the end by saying I'm not advocating anyone always being on the ketogenic diet. I really hope I didn't say that. It may seem like that because when you defend ketones, people can't help but think you're advocating them. I will defend ketones. They've been fantastically maligned. But for those of you who are older and worried about cognition, I unapologetically share with you the data suggesting that ketones are among the most therapeutic things you can do. The evidence that ketogenic diets make people sick, that's simply just social media. It is not fact. Indeed, I would ask whatever preconception you had about ketones coming in, if nothing else, respect me enough as a scientist to know that I'm not going to talk about something unless I am quite confident of the data, not to be swayed by dogma. Yes, please.
Your opinion on two competing ketone defensible lifestyles. One is the intermittent fasting, and the second is the 30-30-30, where you wake up within 30 minutes, you eat 30 grams of protein.
Yeah.
And you go after that.
Okay, yeah, so in fact, good for you. The fact that you know about the 30-30-30 idea, that's a more recent one. Those are not incompatible with each other. So the question about two different...
Stop eating like at 3 in the afternoon.
Yeah. So intermittent fasting is incredibly therapeutic. I know of case reports that get people off their diabetes medications in weeks with just intermittent fasting. My view of intermittent fasting is it can be very powerful, but it can also be very abused. And I'm mindful of the college-aged individuals here who are looking for any way to mask their eating disorder. And intermittent fasting becomes a nice way of kidding themselves into thinking they don't have a problem. So with regards to intermittent fasting, how you end a fast matters substantially more than how long you fast. Let that be written in stone, that if you're just using a fast as a binge purge, you're fasting, and then going bonkers on binging whatever junk you can shove in your mouth. It doesn't matter how long you fasted. You're doing it poorly. How you end the fast matters more than how long you fast. Now, the 30-30-30 principle is very relevant to the aging audience, which is refreshing for me. I'm used to 20-year-olds. It's fun to have more mature individuals here. But the most catabolic state, in other words, the time where we're losing the most muscle is overnight. And you want to then stop that breakdown as early as you can. So my view is actually that after drinking some water, if you're worried about muscle mass loss, have some protein, or if you don't want to eat protein, have some amino acids or something. That would fit the 30-30. Within 30 minutes of waking up, have some branched chain amino acids or something to help stop the muscle loss that's been happening overnight.
What about like a health coach shake or something?
I can't answer that.
A big glass of raw milk.
Oh, yeah, sure. Yeah, please.
Parkinson's disease, you didn't mention that, but I know a number of folks that have had Parkinson's disease. One, my father had Alzheimer's and Parkinson's. And his first thing, somehow he had the glucose and so forth. Does this principle tie over to
Yeah, that's a good question. And let me just remind the audience, thank you all for humoring me as if I'm a neuroscientist. I'm not. I'm a metabolic scientist, so I know a lot about mitochondria and a lot about endocrinology and the influence of hormones, telling fat cells to grow and shrink and telling the body what energy source to use. So please appreciate the limitations in this type of question. But I can answer it, which is that there is evidence to suggest that the dopamine-producing neurons, which are broken with Parkinson's disease, do respond favorably to ketones. So as much as I focused on Alzheimer's disease, there are also case reports documenting significant improvements in Parkinson's with ketones. I hate saying the word like. You 20-year-olds are rubbing off on me. I'm going to start using literally too liberally soon. But you can almost pick any brain disorder, and that's too dramatic. Many of the chronic brain disorders benefit tremendously in response to ketones. In fact, of all the tissues of the body, the tissue that appears to be the most hungry for ketones and benefits the most from them is the brain. We've published reports looking at what ketones do to fat cells, what ketones do to muscle cells, and indeed what ketones do to brain cells. And the brain appears to be the tissue that benefits the most. That's why, among all of the reasons to have a favorable view of ketones, if the brain is the tissue of concern, ketones are your best friend. Yes?
According to the New England Journal of Medicine, now full disclosure, I haven't read the study, but there is a link to it.
Okay.
It says in at least one trial, 22% of patients taking this one Alzheimer's drug, Leqembi, developed brain swelling compared to just 10% in the control group.
What's the mechanism of action of the drug?
Great question. I don't know.
Okay, well, then I won't be able to answer it because I don't know that name.
Okay.
Yeah, sorry. Ray, how many more? 2 more? Let's say 2 more questions.
We haven't lost more than 5 people, so I guess you
Well, you've been sneaking out here. Who's had their hand up? Go ahead, please.
How does this translate into menopause years for women?
Yeah, that's what a great question, actually. So, women have the advantage of dying less from pretty much every disease. Men die more from everything, including suicide, by about 5 times more. But Alzheimer's disease is an exception. So estrogens are very neuroprotective, not only because of a direct effect of the estrogens, estradiol signaling to the neuron to promote longevity of the neuron, but also as estrogens go away, the woman has now become subject to the consequences of her body fat. My students might have heard me joke before that a premenopausal woman is a metabolic superhero. She's bulletproof. She can have much more body fat than her male counterpart, and indeed she should have more by design and be healthier in every metric. And that's because estrogens, just as a teaching point, there's no single hormone called estrogen, so I'm being deliberate in how I'm using the word. Estrogens actually help the female body store fat in a more healthy way and burn fat more. You gals don't know that right now, sitting here, you're burning 40% more fat relative to your body weight than your male counterpart is. But you're also putting more in those fat cells. So all of that is to say the premenopausal woman with high estrogens is extraordinarily protected. With the loss of estrogens comes the loss of that protection. And now that body fat is going to be contributing to fat storage in such a way that it will promote insulin resistance. Now, as I'm answering this, I'm cognizant of the fact that I didn't describe the role of the fat cell in causing insulin resistance. That is just too big of a topic for me to get into. But suffice it to say, how a body stores fat will heavily contribute to insulin resistance or protect. And again, the menopausal woman or the loss of estrogens in the absence of hormone replacement will be subject to the consequences of that fat like her male counterpart is. So an enormous increase in the risk of Alzheimer's disease, of course, because of those 2 factors. The neuroprotective effect of the estrogens directly, and then the protective effect metabolically of the estrogens, which both have gone away. One more question at the back in the auburn shirt, right there in the corner, with the CGM on the back of your arm.
So often you mention how you break a fast is more important than how long you fast. Aside from the binge per cycle, what's a good way to break it? What's a bad way to break it? And why does that matter biochemically?
Okay, great. Yeah, thank you. That's a great answer, a great question. It's going to be a great answer in just a second. I hope. So, yeah, I actually like to look at fasting in 2 different ways. Based in part actually on the work of Dr. George Cahill, one of my legends, one of my heroes. He documented the fasting in the fed state, which influenced or informed my own views, which is there's a difference between a caloric fast and what I call a metabolic fast. A caloric fast is one where insulin has come down, which according to Dr. Cahill defines the fasted state, and it's because you're not eating or drinking anything. No calories, rather. You could be drinking water, but no calories coming in. But then the metabolic fast is where insulin stays down, so the body continues biochemically to behave as if it's in a fasted state, but you're eating calories now, but you're focusing on fats and proteins. So my view is when you end a fast, focus on the most nutrient-dense foods, which is going to be fats and proteins. Did you know that there's no such thing as an essential carbohydrate? that there are such things as essential fats, there are such things as essential proteins. There is no such thing. Don't tell me fiber is essential. It's not. That's why you poo it all out. Now, I'm not saying don't eat carbohydrates. Please do not put words in my mouth. I'm not saying don't eat them, but how bizarre it is that that's become the macronutrient that we are obsessed with when it's the one we literally do not need. Even the Department of Agriculture years ago, I'll loosely quote it, said that the lower limit of dietary carbohydrate is 0 in humans. So how would I recommend ending a fast as just your friendly neighborhood scientist, because I'm not your clinician? I would say focus on the things that matter most, proteins and fats. The worst way to end your fast is to spike your insulin. Because that can, if your fast has been long, it can cause a very harmful scenario called refeeding syndrome, where your blood potassium can go too low. People can die from that. But it also just sets you right back up for the cycle, because the higher you spike your glucose, the higher your insulin will go, and then the lower your glucose will go. And that rebound hypoglycemia will drive hunger sooner than it should. If a human eats the same 2 meals, calorically the same, but one has lower carbohydrates and one has higher carbohydrates, the higher carb version of the isocaloric meal will drive hunger much sooner, rather than the lower carb version where insulin has stayed low. I would say let your nutrients work in your favor. So the best way to end a fast would be those 3 pillars that I showed you earlier. Thank you all so much for coming and listening. I appreciate it. Thank you. Thank you so much.